largely beyond our control. And so people started to ask, well, can we just use dCas9 fused to additional domains to mutate the genome in a specific way? And this is a process that happens normally during B cell development and antibody maturation in all of us. And so what David Liu's lab and Mike Batsik's lab showed last year was that you could fuse dCas9 to these cytidine deaminases, either from rat or the human AID protein that's involved in antibody maturation, and then mutate the genome without forming double-stranded breaks. So these are targeted mutations that are induced in an RNA-guided way, and you predominantly, either depending on the fusion proteins you use, get C to T mutations, or in Mike's paper, they showed that you can get C to N mutations. And so this is, I think, an additional application of dCas9 that people seem to be quite excited about, and there was a sort of an explosion of papers in Nature Biotech a couple weeks ago where people used one of these two systems to implement DNA editing without inducing double-stranded breaks in a variety of types of organisms, and showed a lot of applications. David Liu's initial work showed that you can also fuse on additional domains to increase your rate of editing. So they fused on a uracil DNA glycosylase inhibitor onto dCas9, and this increases the conversion of mutated base pairs, and that's detailed in the paper. I won't go through it for time, but both David and Mike have shown that this can be used to study anti-cancer drugs, or to, in theory, in a very specific way, mutate disease alleles from their pathogenic sequence back to a wild-type sequence, which could have therapeutic applications. And so I'll end there and just say I hope that I've convinced you that as awesome as Cas9 is, there is a place for thinking even beyond Cas9's canonical activity and using repurposed forms of CRISPR enzymes to do additional things in bacteria, yeast, and human cells. And we're pretty excited about that. So, thanks.
Excited to discuss my favorite topic in the whole world, which is